Welcome everyone to today's webinar. I'm Ayla Ellison with Becker's Hospital Review. We will begin today's webinar with a presentation and we'll have time at the end of the hour for a question and answer session. You can submit any questions you have throughout the webinar by typing them into the Q&A box you see on your screen. We look forward to hearing your questions. This session is being recorded and will be available after the event. You can use the same link you used to log into today's webinar to access the recording. At this time, it's my pleasure to start today's webinar by introducing our presenter. Boyle Chang, PhD, holds an appointment at the rank of full professor in the Department of Neurosurgery at Drexel University, Allegheny General Hospital. He is the Director of Research for the Neuroscience Institute at Allegheny Health Network. Dr. Chang also maintains an adjunct appointment in the Department of Biomedical Engineering at Carnegie Mellon University, serving as an advisor to the graduate students in the practicum course and teaching the medical devices class. His grant-funded research has included the evaluation of anterior cervical disc replacements and novel biomaterials in a sheet model, biomechanical studies of a biased angle posterior cervical system, facet motion preservation technologies, as well as dynamic and physical modeling based on a specimen-specific imaging and kinematic studies. At this time, I am pleased to turn the floor over to Dr. Chang to begin today's presentation.
Ayla, thank you very much. Well, welcome everybody. I also appreciate your time and wanted to thank each and every one of you as we discuss a very exciting topic. This is a robosis, which is an electrospun fiber technology. Before we get started, these are three cars that not only adore the walls of many young high schoolers, but I'm sure they fill the minds and garages of many people in the audience, or at least in our fantasy garages. One thing they have all in common is the fact that they are a mid-engine design. And after 30 years of development, Corvette has also decided to join this particular design attribute and join the class of elite supercars that are currently available. 2020 was a great year for many reasons, and we look at the attributes that the mid-engine brings to the Vette and the standard performance package. One of the exciting things about it, it has really led to an amazing track car now and an everyday driver. But what I really want to talk about is how this translates into the orthobiologic world. When we start to look at these calcium-phase products, there's a lot of them, but this is a platform technology that I think robosis has revolutionized. Primarily, this is due to how it's manufacturing. When you look at the electrospun fiber technology and what they're able to do with the manufacturing process, it tells you a lot, and through a lot of scientific evidence we're going to present to you today, you're going to see why, from a cell biology perspective and some of the in vivo studies, we are excited about this calcium phosphate product. When you look at the C4 design of the Corvette, this was always been a great car and a great American-made car, and for many years we had come to accept that as the design. And with the advent of the mid-engine car, we're building upon a great technology and making it better, just like the Vette is in 2020. So really excited about the potential and the technology associated with it. Before we start, we have to look at the regulatory classification and how that's perceived. When you look at this specific class of orthobiologic, they break it down into a bioactive ceramic. And within that bioactive ceramic, there are three separate groups. Some of them are considered bioinert, some are considered bioactive. This happens to be a bioresorbable product within these bioceramics. And what makes them particularly interesting is the fact that over time, as a part of the bone remodeling process, they are able to then grow a sufficient amount of bone and a lot of bone in specific application. And in this category, we see that HA and beta TCP are two bioceramics that dominate this specific space and will be continued to be perceived as a great product with those that incorporate that type of ceramic. When we look at orthobiologics, we see that the application is it's broad ranging in orthopedics. It's not limited to just joint replacement alone. We see applications for spine, we see applications in trauma, and we see applications in sports medicine. These are all very important segments of the orthobiologic market. But as within any year, as we continue to see the growth of this orthobiologic market, we've had to take a back seat due to the pandemic. And based on these projections from the market report, in fact, the impact of COVID-19 has, in fact, affected the market and obviously affected the procedures. These are aspects that have probably affected everyone's practice, everyone's institution, and certainly the market in general. These are things we expect to return, however, in 2021, and we expect to continue growth in subsequent years. So that's why this technology is particularly important for the future. As we look at this growth market, we look at all the competition in this space. It is certainly a crowded space, and trying to understand what differentiates these products ultimately makes us understand not only the product itself, but the best application for these products and which patients would benefit most. So if we look at the essential components of bone healing, we notice that there are three elements that are absolutely necessary. That's the matrix component, or the substrate. This is where the cellular activity would make themselves at home, so to speak. And in combination, they secrete a number of growth factors that start that bony cascade. It's this trifecta that's important ultimately for bone healing. And regardless of the combination or the order, the fact that you have all three is essential to bone healing. This reminds me of some of the things that we thought of that came in groups as well. Growing up in a small rural town in the eastern plains of Colorado, there's a lot of people that drove Chevy, drank Budweiser, and smoked Marlboro. But just as cool were those guys that were driving Ford, drank Coors, and smoked Camels. Along came Dodge, Miller, and we don't want to forget the Lucky Strike. And I know that these smoking analogies are way passé. It's not something that we advocate any longer, but it's just a sign of the times and what I saw growing up. But it reminds you of the fact that all these things, they come in triplicates, and it's something that we associate. But as necessary, it comes in threes, particularly for bone healing. And that's what robosis really brings to the table. It has a number of components essential for that bone healing process when it serves as the matrix material or that substrate. Particularly attractive, as we point out, is the bioceramic component, the orthobiologic, which happens to be beta-tricalcium phosphate. That is on the outside of the fiber itself. But it has a silicon dovatorite, which is a form of calcium carbonate, and it's built on a resorbable scaffold, a PLGA, something that we know and how it breaks down and how the body metabolizes that particular polymer. What you see is a picture on the left. This is the gross form of it on the top, and under electron microscopy, you can see the fibrous nature, and you get a sense of that scale when we look at it under electron microscopy. That particular aspect of it leads to a very useful clinical application, and here are a couple of handling characteristics that make it a great product. Number one, it has a high level of absorbency. When in the presence of a liquid, it has this wicking capability due to the design of the fiber itself. It also has an increase in volume for cellular material attachment as a graft site. And ultimately, it's because of this fiber organization that allows it the spatial organization that is not commonly found amongst other orthobiologics. Again, going back to some of the microstructural aspects of it, if you look at the scale, I wish I had a pointer, but if you look closely on the right-hand side, you'll see this particular scale, and this describes the micron level features that you will find on the specific fiber itself. So these specific fibers, in combination with that beta-tricalcium phosphate and the other essential elements, make up a product that I think is very conducive and makes for an excellent matrix material for bone on growth. If we look at the picture on the left, there's a marker. That little white bar in the middle on the picture on the left is 200 microns. What that should describe to you is that a fiber itself is on the order of approximately 10 microns, and this is something that is not achievable by any other manufacturing process. And again, makes this product unique because of the microspun fiber. But is it a good idea? Clinically, having this particular product, what does that afford us specifically for the patient, and what do we know about it? Well, it is a synthetic. It has labeled indication as a bone void filler. It has a very great osteoconductive scaffold that has this cone-like structure that makes it very attractive for not only for the initial cell activity and attraction of those initial osteoblasts into the actual substrate, but it also promotes the proliferation at a faster rate of incorporation. And we're going to see how our cell evidence supports that aspect of it, as does our in vivo studies that have been performed on this particular material. All of this we attribute, again, to the electrospinning aspect of the manufacturing. It's the production of these thin fibers, which would not otherwise be available if we were trying to process it in any other method. So this makes for an exciting array of possibilities when we have that manufacturing tool capable to produce this particular product. So we'll take a quick look at the physical properties associated with robosis. This includes the porosity. We'll also discuss the surface area, as well as the compression recovery characteristics. And this all goes into handling aspects, which are essential for the physicians, specifically the surgeons that would be using this in an application. We also want to talk about the biocompatibility. When we look at cell proliferation and cell morphology, that tells us a lot about how this will interact with the host tissue environment. So when it's implanted, what does this actually do, both on the cell level, and also what evidence do we have of that in an animal model? And finally, let's talk about the cellular activity specific to the bone healing. We'll look at osteoblastic differentiation, as well as the other osteoblastic activity that indicate that it, in fact, this goes on to maturation and ultimately mineralization of the particular host graft. So when we talk about the structural properties, one very important aspect is the scaffold and the form that it takes. A good way to characterize it is through micro-CT analysis. One of the first things we wanted to do with it is to understand the porosity and specifically how it was able to achieve that porosity. Instead of having specific pores, this porosity is associated with the fiber to the overall volume that it occupies. So in a general state, we're able to characterize that by volume. And when we compare it to some of the other competitors, we see that the fiber format to compare to the overall volume is some of the lowest available. As a matter of fact, that fiber volume to the total volume is on the order of what we see in trabeculation, particularly for L3, for the vertebrae and the cancellous bone found in L3. You'll notice that on the second row, robosis is characterized in the second column as bone volume to total volume. And that comes in at about 9.83, where cancellous bone registered as a percentage of about 9.3. So this is excellent in terms of how much void you have in comparison to the fiber. It gives it plenty of room. And more importantly, the next column describes a very high surface area to volume ratio. This indicates that for the given fiber, there's plenty of surface for cells to attach to, given the same amount of material, the same volume of material. And then ultimately, that trabecular thickness is characterized, again, by the material that we had just shown and looked under electron microscopy. So those aspects under micro-CT analysis give us good insight as to why that substrate is so friendly and so familiar for cells to ultimately end up and reside when they go on to remodel and to go on to heal in these specific applications. We're quite excited about that and think that that aspect is worth noting from a physical attribute standpoint. When we look at the compression characteristics, they are also equally valuable. Not only when is it hydrated becomes moldable, but it affords a high bone-to-graft contact that makes it very conducive for bone void filling. We want to make sure that it will conform to the space that it's intended to fill, and that's what the moldability offers when robosis is actually rehydrated. The compression recovery characteristic shows that on average, there's a recovery of about 54%, meaning that once you squeeze the particular material, it will recoil back to the amount that will fill a void. And in the era of expandable cages and expandable areas, those are really important aspects for your graft material to fill once they're inside the inner body space and the cage itself is deployed and extended to the height desirable by the physician in that particular case. So what we're showing here is there's a preloaded condition, and as it is compressed, which is often done on the back table as a step prior to insertion and implantation of an inner body cage, it will recover not only immediately after the load is removed, but in the inner body space, it expands to fill that void, and that is an excellent property that we like to see related to these graft materials in that space. All this leads to, why is that important? Why is porosity important? Why is this specific density and the surface area important? Well, ultimately it comes back down to what we were talking about in terms of the trifecta necessary for bone healing. There needs to be a good host environment for the osteoblastic activity to occur. So we start on the right hand of this right-handed picture. We saw that these messenger signaling cells are attracted, and as they differentiate and become these osteoprogenitor cells, then the pre-osteoblastic activity forms into the osteoblasts that become the surface aspect of recovery in terms of remodeling and healing of that inner body space. But this is why bone is such a great material. It is one of the perfect healing types of tissues in the body. It's because it's the other component that is essentially as important as the osteoblastic activity, and that would be the osteoclastic activity. So as you form bone, your body also seeks to repair bone, and that repair aspect of it, the osteoclastic activity and the resorption aspect of it, leads to this eventual cycle that repeats itself over and over until such time that that bone has formed and healed in a perfect manner. It's the continuous nature of the osteoblastic activity in combination with the osteoclastic activity that make bone such a wonderful material, and why robosis is such a great matrix material to serve as a host environment for that cell activity. One of the ways we have confirmed this aspect of it is through the assays associated with cell growth, cell proliferation. We also look at cell maturation and development of the extracellular matrix. And in the third phase, as the maturation process goes on to mineralization, we have all different techniques to ascertain exactly how well this material performs at each one of these separate phases. The cell differentiation component can be picked up by the alkaline phosphatase assay that we perform. It can also be done by PCRs and other ELISA. So we want to look at some of those attributes and how we were able to do that. Specific cell lines that we chose were the MC3T3 cells along with the C2C12 cells. There are different types of cell lines. The MC3T3 cells are the pre-osteoblastic line, where the C2C12, a common line to use, are a myoblast origin. So those specific lines were used when we looked at the proliferation stage. When we want to ascertain biocompatibility, the ability not only for cells to differentiate, but then to become more mature and develop that extracellular matrix, there's a number of assays that will help you establish biocompatibility by looking at the ATP alkaline phosphatase response. And those particular assays tell us a lot about that maturation process. And what we see at the 24-hour and 72-hour time point, robosis, in fact, particularly in the 72-hour, excels compared to all the other products that were tested in these static weld plates. So for this cell culture, we were excited to see that in 72 hours, at that midterm point, that cell maturation, that extracellular matrix did indeed form and formed very well within the culture that we tested in within these wells. We were excited to see the comparison relative to graft-on, intergrow, and stay graft. If we wanted to take a look at the optical microscopy aspect of it, we could put it under a scope, and under that fluorescence, you can really see all the activity of robosis, which is in your upper left-hand corner of these 4 particular products. These other pictures are taken at the same mag and also at the same time points, and they don't show the same amount of alkaline phosphatase activity for the same period. At 72 hours, that's where if we went back to the graph, you would see an extraordinary amount of cellular activity. And again, we were delighted to see how much of the green, the fluorescence that we saw, indicating the alkaline phosphatase activity at that time point. What you happen to also see is a blue overlay, indicators of cell activity on top of not only the fibrous material, but also within the ECM itself. So excited to see that activity, and quite pleased that at 72 hours, there's a lot of activity that we could associate with maturation of that cell line. Okay, so the 48-hour and the 72-hour activity, those are all aspects of it that we've described it. Again, happy to see that, and this is, again, something that from that expression tells us a lot about that process of maturation. And when we put it under optical microscopy, what we see here is that those areas in between the fiber, under bright field optical microscopy, we can see the ECM forming, we can see the activity associated with it. And again, this is going to be the second row in figure C and D, and you can see the activity in between. I know they're a little small, I apologize for that, but the activity that is between the fiber is something that we are excited both at the 48 and the 72-hour time point compared to a lot of the other activities that we see in some of the competitive materials. So really excited about the maturation process. We also are able to use other techniques, including DAPI staining, which highlights the cell itself, the cell nuclei. And then also with the green, we're able to look at the cytoskeleton aspect of it and assess the cellular morphology with it. So these cultures were taken out much longer. These were at the 7 and the 14-day standpoint. When we look at the osteoblastic maturation component, we start to see that there is, in fact, quite a bit of activity at those particular time points, indicating that in the clinical setting, they should be also very successful. So again, although hard to tell just on this one cell culture study alone, but there is a suggestion that the progression of this particular material and at the rate at which it becomes mature really does lay down a better bed, a better extracellular matrix early on, leading to faster maturation and ultimately to faster bone healing that hopefully would translate into the study subject as well. So coming back to it, again, on your screens, you should be able to see the nuclei being stained specific to the fiber. And this is where we are excited to see that the surface area does come into effect. What we like about robosis at the 7-day time point is the amount of cellular activity specific to each of the fibers. But more importantly, at day 14, what we talked about is, again, if we look at the blue nuclei that have been stained by the DAPI staining, if we look at the green and the cellular activity associated with the ECM, both combinations on the surface of the fiber and between the fibers, you'll see in that upper right-hand corner, really illustrate how strong a presence not only of the osteoblastic cell lines that you see at that time point, but also the cellular activity and the extracellular matrix that are present within the same time frame. In comparison, the competitive materials just don't have the cell activity at either the 7-day or the 14-day, illustrated by the 2 figures on the bottom. I'm using a graph-on substrate. So, is the C8 actually better than the C7? That's literally the question, right? So does moving the engine to a mid-engine format, does that actually make a difference? And given the same trim level, given the same package, Car and Driver actually took these Corvettes out on the track, and the real-world test, they wanted to see if, in fact, they did perform differently. And given that the C8 had better lap times, had better braking, going into the corners and ultimately better acceleration and harder out of the corners, it resulted in faster straightaway times, a 3-mile-an-hour advantage on the C8. That's really an exciting attribute when you look at what a design change ultimately translates into the real-world aspect of it. And similarly, we would hope that these changes to the design of calcium phosphate-based material, again through electrospinning technology, will helpfully, in the real-world analysis, show us that does result and translate into much better bone healing. This is where I get really excited. When you look at the 21-day outcome of these assays, now we're talking about the limits of what we can do in cell culture study. But this is really where the bang for the buck really pays off for benchtop studies. In this phase, if you can show that mineralization is better at this long of a time point, that really becomes truly exciting. And that's indeed what we found when we look at the calcium assay with these MC3T3 cell lines at 21 days. An important aspect of mineralization, after maturation of the cells, we have this mineralization, and that's how we know that, in fact, that cell activity really does heal and turn into the bone that we needed to form, and that mature bone, ultimately evident by the mineralization phase, to be important in what we think is the bone healing process. So in this particular graph, at our 21-day calcium assay, we see that in comparison to its competitors, by far, the maturation of robosis was significantly higher than the competitive products we tested in static weld plates at 21 days. The top right-hand picture is what we wanted to show you. When you look at the osteocalcin and the staining between the fibers, in fact, that is picking up on the mineralized component, not just of the fiber, but of the extracellular matrix and the components that are mineralized throughout that entire bottom of that specific weld. In comparison to the picture on the bottom, where you just don't see the same level of mineralization, the same level of activity. And when we go to the other competitors, when you look at robosis at 21 days, it's just amazing how much of it is red, how much of it at that particular well, and in those particular wells and between those fibers, how much of it is being mineralized. Something quite exciting. When we look at other competitors, we see that there's patches that aren't filled in. They're not nearly as saturated in mineralization, all indicating that robosis, in fact, does lay down the amount of mineralization that's quite exciting. Head-to-head comparisons are still important. We want to know in an in vivo environment, whether it's an animal model or in a clinical trial, what does that look like compared not only to itself and its previous generations, but in competition against some of the best materials out there. How do we establish that? There are specific ASTM guidelines that instruct us on what these environments would best be to compare, whether they're in separate cohort studies or whether in the same specimen and the same animal. And specifically on this ASTM guideline, this is 3207, looking at a rabbit model. You can use these specific rabbits in a posterior lateral onlay fusion to see how they compare against some of the competition. So these types of studies tell us in the in vivo real-world models what happens to bone healing and bone modeling. This is work that Dr. Fredericks was kind enough to share with us. He's at the University of Iowa. Does a fantastic job. Has a ton of these rabbits were submitted actually in support of the regulatory process in clearing these materials. So fantastic work that he has shown, and one of the best take-home messages that he could show you was what happens to bone formation under micro-CT at specific time points. So at the 12-week time point, in combination with iliac crest bone graft, this fusion mass, the arthrodesis that you see as an on-lane fusion, was phenomenal. This is also verified by other means, including palpation of the harvested spine and by many other methods. But because of the micro-CT, I think this is an excellent indication of how solid that fusion mass is, even in a posterior lateral onlay type of model. So fantastic work by Dr. Fredericks and his team at the University of Iowa, who have researched this product extensively in a rabbit model. So a couple of take-home points. It's clear working with robosis that the material presses an inherent charge. This is, again, a function of electrospun technology. That in itself is attractive for many reasons, no pun intended. When we look at the surface and the benefits of having a thin fiber with a large amount of surface area, this really is attractive, not only on the molecular level, but on the cellular level. Those cellular interactions that we saw on the cell bio side of the studies really represent an excellent scaffold. And we feel that with more time, understanding that surface charge and the attraction that it creates with the cell will lead to a lot of a better understanding of the optimal material and designs necessary for these materials to be attractive for the best environment for a cell bone implant interface healing due to cell activity. Through the proliferation studies that we've already done, the mineralization and the maturation aspect of it, we feel that robosis has demonstrated quite a unique material that will benefit the patient with faster healing at specific time points. So in conclusion, we find that robosis and the electrospun fiber technology provides excellent handling properties that we talked about, including compression and recoil recovery. We also feel that robosis allows for rapid proliferation of osteoblasts, that these cells proliferate along the fibers before beginning to fill the space between the adjacent fibers. And then ultimately, robosis supports osteoblastic activity as evident by the alkaline phosphatase expression, extracellular matrix formation, and ultimately the mineralization that we saw through the Osteocalcin in the last slides. Thank you all for your time. It's been a privilege of mine to work with Kyocera on the robosis product. It's been exciting to see this new technology, and excited to hear your thoughts on this particular technology.
Thank you so much, Dr. Chang, for that fantastic presentation. And we'll now begin today's question and answer session. So please submit any questions you have by typing them into the Q&A box you see on your screen. And we'll try to get through as many questions as we have time for today. So looking at the questions that have already come in during the presentation, we'll go ahead and get started with those. This first person is asking, does this have implications for osteoporosis?
That's a great question. When we look at a lot of the pharmacological agents currently available for the treatment of osteoporosis, that tells you the importance of having the ability to heal bone. This is not the same as a pharmacological agent. So for surgical interventions, we want to make sure that they are given the best local environment to heal that specific wound. When we look at the bone void filler indication, yes, this is a great material for that aspect. We have to remember that's only at the local level. So excellent for that purpose, excellent to support that specific local environment. But when you start to consider things like Forteo and all those other things that work on the endocrine system in general, they approach it from a different route. And having that aspect of it is still very important, but having the best environment at the local level is essential for the success, particularly when we look at spine or orthopedic applications. You want that healing to occur at the local level, and that's exactly where robosis excels.
Thank you so much, Dr. Chang. And the next question that came in is someone is asking, are clinical trials underway on robosis?
Yes, there are retrospective aspects that are being collected right now. We have a number of sites out there that are very much interested in looking at their cohort. We are looking together to put together a potential future site, but it's too early to reveal what that will actually entail at this point. But excellent question. That is always the number one evidence that the device will perform as you think it will. We can do all the studies that we want, but ultimately it's going to come down to the performance in human subjects that will give us that level 1 evidence, and we're all looking for the same thing. So designs are in the works.
Thank you so much. And the next person is asking, what is its regulatory status?
It is currently cleared at the agency, at the FDA. As I said, Dr. Frederick's work went to support some of the early submissions, and it is specifically approved. Sorry, that would be a 510K clearance, so it's cleared for commercialization in the U.S.
Thank you so much. We've had several questions come through, so I'm just going to continue to work through those. The next question is, would you entertain the possibility of combining the material with a bone anabolic agent?
Absolutely. So going back to one of the earlier slides, and then maybe I can bring that up quickly, it's that combination, a matrix material as osteoconductive as it can be, the essential aspect is to make sure that all three components are there. So if there are anabolic agents that help attract specific cell activity, then you have all three. As we stated early on, it doesn't really matter in what order they show up, as long as they three are present. That trinity ultimately is essential for the success of bone healing. Trying to get that slide back to you. There we go, thank you. So when we look at this slide and we put an anabolic agent on top of that matrix, the fact that it's osteoconductive is excellent, but the cellular component is still needed. Let's not forget that aspect of it. So whatever combination we have, it's always a good start. We could have gone the other way as well. If you have just a matrix material and you have some progenitor cell line, if you have so I hate to say stem cell derived cell lines, but in combination, that would work the other way and attract and secrete that chain of proteins that would cause that cascade and ultimately for bony fusion to still occur. So all aspects of that is important, and regardless of the order, the key is to having the three components present.
Thank you, Dr. Chang, and for pulling up the slide. That's helpful to be able to visualize that as you're explaining it. We've had a few more questions come through, and the next question that came through is, have there been any maxillofacial application studies performed up to this point?
Great question. I always feel like we take from MaxFace, and there are applications in that they have various applications and also have the same type of need. And when we look at orthobiologics and what they've done in that space, and even the pioneering of a lot of the modern day materials used in design, I think that MaxFace applications have a good understanding, and it's based on all the same principles, right? The loading conditions may be a little bit different, the size and the volumes may be a little bit different, but they all work on the same principle, and that's healing of bone. So whether this is in a MaxFace application or in a long bone femur fracture model, it all goes to the same principles. Those orthopedic principles hold, whether it's in the face, in the jaw, or in the leg. So we rely on their experience and their expertise, and they tell us the same thing about that application as we do in what we find in the spine or a distal femur type of bone void fill that we do.
Thank you. And we've had another question come through, and this person is asking, what is the indication for robosis? Does it have to be used with blood, BMA, autograft, etc.?
I'm going to leave it on this slide because, again, I think it's really important. So when we look at blood, the picture that we show is that it has excellent absorption characteristics, and that's due not only to the surface aspect of it, but the ability to wick through those the void within that space. Even under compression, it will still continue to wick liquid material up. Blood isn't as important as, again, getting to the cellular component. If we can get to those messenger signaling cells, the MSCs that are found in cancellous bone, if we can get to the bone mineral aspirate aspect of it, that's more important than blood per se. Blood, as interesting as it is, it's really a proxy for what we need, and that would be the cellular components And those are the aspects that we would want to make sure are retained within that matrix when put in combination with bone mineral aspirate. This is something that you have to harvest for spine procedures. It's easier to harvest out of the iliac crest or the local environment, but it's essential that we get to those MSCs in order to see that those cells on the matrix in order, again, to start that growth factor cascade that will trigger the rest of the healing and resorption characteristics that we know occurs with osteoblastic and osteoclastic activity.
Thank you so much, Dr. Chang. And the next question that we had come through for you, this person is asking, with the surface charge, is there any antibiotic potential?
Areas that are all of great interest to a lot of different people, so I can say that ongoing research is exactly in that space. Again, this is a benefit, an upside that we get from the electrospinning technology. We're exploring that aspect of it now. Not only is that a great thought in terms of the antibacterial, antimicrobial aspect of it, but potentially what can we do for cell attraction and help cell adhesion based on the charge associated with the manufacturing process. So things that we're exploring right now, not able to comment specifics related to the research, but we hope to find something very exciting in that space shortly here.
Thank you so much. And we'll go ahead and move on to the next question. We have time for a few more. So this person says, I use expandable cages for some of my patients, and I worry about graft contact with the end place after expansion. Does the compression recovery of this product make it effective for use with expandable cages?
Yeah, and that's an excellent question. That's why we did some of the recoil characterization of the device. With the compressibility and thus expandability with these cages, it's an essential element. And based on the compression recovery that we witnessed in that trial, we do feel that you are able to not only deploy it in a compressed format, but as that cage expands, the graft will expand with it. Now, there's still a utility in adding additional graft material. I know that a lot of the expandable cages do allow for additional material to be added after final placement of the inner body cage, but ultimately the ability for the graft itself to expand to fill that volume is something that we witnessed and has been actually documented in other studies as well. So not only was that confirmed within our lab, but there are other studies to support that, and that 53% expansion was something published from another lab. So great findings, excellent application. So expandable cages, a very important part of the triage available to surgeons, and I think this graft material in combination supports for an excellent fusion recovery.
Thank you. And the next question that came through, it goes back to something we already touched on in the questions, but or in the previous questions. But this person's asking, based on this data, is bone marrow aspirate the ideal growth factor to use in combination with this product? And what about infuse?
Yeah, so is it ideal? It depends on your circumstance. Every institution is a little bit different. There are cost pressures that are different from institution to institution, availability from institution to institution. Bone mineral aspirate is available to almost everybody. There's not a single surgeon out there that wouldn't be able to harvest BMA. Do they harvest enough? That may be a question. The availability at a specific site, do they have to open up multiple sites? Now that becomes a question, right? So although it is something that is accessible, is there enough of it? So in those large defects, that's something that you would consider an extender, something that may be a synthetic. This is where robosis really comes in. So in combination, yes, it's an excellent material. Had we had a perfect world and we could expand those specific MSCs within the bone mineral aspirate and then reimplant that back into the patient, that would be more ideal. You would get the cell density, you would get the cell count that would ensure really good coverage and really good fill of that volume with the appropriate cell activity. So although the harvest is important, I still think if there were more of them, if there were ways to implant additional stem cells to support that, that would be great. Is infused an ideal material? Infused is a protein, as there are other bone growth proteins out there as well. We know of others that have been used in this space, or combinations and cocktails of those. So there's many ways to solve that problem. And when we look at this slide, again, it comes back, are you filling it with the cell component on top of that matrix, or are you attracting the growth factors and seeding the matrix with those proteins that make it the best environment? Everybody has an opinion on this, but again, I would say that having 2 of the 3 to start is excellent, but you need all 3 ultimately for that patient to be successful, and the body does a great job of filling in the 3rd, whether it be a protein or whether it be the necessary cells.
Thank you so much, Dr. Chang. And it looks like we have time for a few more questions before the top of the hour. So I want to go ahead and move on to our next question, which is, does the electrospinning process leave a surface charge on the fibers, and does that help with cell attraction?
We believe it does, and that's the exciting part, that when we open the package, we notice exactly that. The packaging maintains that charge, and it's a very attractive feature for us. We think that from the hydrophilicity aspect of it, we also think from a cell attraction standpoint, having a slight charge to the product is interesting, not only for cell activity. We know that electrotherapies depend on that specific principle by itself. We're not saying that's anywhere near the level of what these stimulators do, but it is an important consideration at the cell level to say that there is a charge associated with it and would promote cell attraction. So we do think that there is benefit from that electrospinning process as a byproduct of the manufacturing.
Thank you. And moving on to our last question for today, this person is asking, how would you compare this to DBM-based products?
Demineralized bone matrices have been around for a long time, and we compared robosis to the granddaddy of them all when we look at graft-on, right? And so I think that DBMs are, in a lot of ways, just another matrix. I don't want to belittle anybody's product out there, but the DBMs are well understood. They have a collagenous component. There's an component that serves as a matrix. There's a lot of other competitors to DBMs. For example, collagen matrices by themselves or in combination with other factors perform about the same. We see that activity associated with type 1 collagen, the activity associated with DBMs, at the end of the day, you're depending much more heavily on the surrounding host tissue. And by that, I mean that you need the cells to, again, be attracted to the substrate. Nothing wrong with it as a matrix material, but it doesn't have the same attraction, the same affinity, if you will, that cells have to electrospun fiber. It comes down to surface volume, size, the amount of void. All those things are essential in the formation and the structural aspects of robosis that make it very, very attractive. Where a lot of the DBM putties and the gels, although great at filling a void, don't have the active components or to attract the active components that really give it a good start for remodeling.
Thank you so much, Dr. Chang, for answering all of these questions that have come through today. I know there have been many. But that's all the time that we have for questions, but I want to once again thank you, Dr. Chang, for your excellent presentation, and thank you, Sarah, for sponsoring today's webinar. I hope everyone enjoys the rest of your day, and we look forward to having you join us for future webinars.
Thank you. Thank you all.